Good morning. We're very happy this morning to have a biostatistician speaking to us straight from George Washington University, Dr. Mary Foulkes. And I apologize to her ahead of time that we're conflicting with ENAR. So all the biostatisticians, I guess, are down in Washington while we're up here. But anyway, I can promise you have a good and serious audience here, and more, since it's early in the morning, will be coming in. So, but Dr. Foulkes, I want to tell you a little bit about her because she has a good background for us and for the topic today, which is non-inferiority and other regulatory issues. So, Dr. Foulkes got her PhD in biostatistics, from University of North Carolina in Chapel Hill. After that, she went to NIH, and she worked at a number of the institutes on different projects, including National Institute of Neurologic, NINDS, Neurologic Disorders, and stroke, that's it, NIAID, and the Office of Research on Women's Health. She also spent about 9 years at the FDA, and there she was the director of the Office of Biostatistics and Epidemiology at the Center for Biologics Evaluation and Research, CBER. She was there, 8 or 9 years, and now is at George Washington University. There she's engaged in clinical trials research and other research. She's co-PI of the Data Coordinating Center of the Lung HIV Microbiome Project. She's working on a project on investing in the scientific workforce, the analysis and modeling project, and also is working as a statistician on a project related to diabetes prevention. So perhaps the most important thing to all of us is that Dr. Foulkes teaches 2 courses here. They're online during the term. She just finished up a brand new one on non-inferiority and equivalence trials. That was 3rd term. And will be offered this summer as well for in person for those of you who didn't get to take the online version. I see at least one person who did. And then she also teaches in alternate years an online course on biostatistics and medical product regulation. And during the summer, and that would be next summer, teaches it in person. Those courses are taught jointly with Dr. Simon Day, who is based in England, but comes over for the live chats and the summer courses. So without further ado, I'll introduce you to Dr. Foulkes, who's going to talk about non-inferiority and other regulatory issues.
Thank you, Kay. Thank you very much for the invitation to be here today. I got to stand. Let me try over here and see if this is better. Okay. So what we're going to talk about today is non-inferiority and other regulatory issues. And before I just launch right into that, you're all here because you're curious about non-inferiority, I assume. But you also know about non-inferiority already. Did you know that? You do know about non-inferiority already. Has anybody washed their hair this morning with Suave shampoo? Okay. Did you read the label on the Suave shampoo? I'll tell you what it says, because I didn't bring my slide that says Suave shampoo. It says, just as good as Paul Mitchell. Now, what does that mean to you, the consumer? Just as good as Paul Mitchell.
Especially if you don't know that Paul Mitchell is a brand of shampoo.
Paul Mitchell is a brand of shampoo, yes. You have to know that, yes.
It's also very important off the market.
Yes. Maybe it would be clearer if it said just as good as Pantene or just as good as Head and Shoulders. So what does it mean to you as a consumer that it says just as good as something else? That it's non-inferior. It's non-inferior in what sense? The result. Maybe it's the result. Maybe it's the cost. Maybe it's a lot of other things. So you know about non-inferiority. If you go to the same pharmacy that I go to, the same grocery store that I go to, they have a house brand that's called Equaline. And they sell everything under the sun, under the Equaline label. They even sell the other example that I was going to bring that I left in my car is wet wipes. And it says just as good as Pond's on the label. So when you go to the grocery store, look at the house brand of Cheerios and see what it says on the label. It says just as good as Cheerios, and it doesn't cost as much as Cheerios. So you already know about non-inferiority. The little extra that I'm going to introduce you to today is the regulatory side of life. Because you could sell Cheerios or you can sell wet wipes, and you don't have to deal with the regulatory side of life that a new drug might have to deal with. So that's the sort of thing that we're going to cover very briefly. And I can't get this to work. Let's see. There. All right. So this is an outline of what I'm going to cover. I'm going to cover some of the basics that apply, that you already know, that apply to non-inferiority and equivalence trials, as well as any other kind of trial that you're dealing with, but actually maybe sort of more exacerbated in the context of non-inferiority trials. I'm going to describe non-inferiority trials in some detail, talk about the impact of missing data, talk specifically about trial conduct and reporting, particularly reporting as it relates to non-inferiority trials, and a few other issues. Let's see if I can get it to work from this side. No. All right. Okay. So in terms of the kinds of things that you already know that apply to non-inferiority trials, these are the sorts of things that you learned in your basic biostatistics course and apply to all trials, but apply really strongly to non-inferiority and equivalence trials. Independence. Now, we always sort of pay lip service to independence, but in a regulatory context, you really have to demonstrate independence. You can't just claim, well, we set up this firewall between the people who are blinded and the people who are unblinded. We set up this randomization process so that they will not know what the next random assignment is. We did all sorts of things in terms of the way we conducted our trial to improve the independence of the observations and to allow us to assume that independence as we go along. In a regulatory context, you basically have to really prove that you've done that and really prove that it worked and it was successful. Reproducibility. The whole regulatory process assumes reproducibility, that what you outline in your protocol, what you outline in your trial plan, what you outline in your statistical analysis plan can be reproduced and will produce very similar results as going on. So this is a real linchpin of the regulatory process. And then there's experimental design, and we're going to talk a little bit more about that as we go along. But you have learned your experimental design. You've learned case control studies. You've learned cohort studies. You've learned randomized control trials. And there are a lot of permutations of the experimental design which need to be explained in a regulatory context very, very rigorously. And hopefully you'll see that as we go along. And then there's the analysis. There are lots of ways to analyze a clinical trial. But in a regulatory context, you basically lay out your analysis plan. Do you have a question? Yeah, go ahead.
Because I think you use the term reproducibility in a way that is a little bit different.
Yeah.
As opposed to precision. I think, wouldn't it be better to say verification? That you basically have documentation of the procedures.
Yes.
Somebody can, repeat
Yes.
the results that they
Mm-hmm.
get themselves rather than doing the experiment twice.
Yes, you're absolutely right. It is actually reproducibility and verification, both. But what I talked about was more verification. You're absolutely right. Yeah. So back to the analysis. You have spelled out your analysis plan a priori. This is a major difference between the regulatory world and the non-regulatory world. You can do a study, do an analysis. If you don't like the analysis, do a different analysis and publish that different analysis and never discuss the first analysis that you did in a non-regulatory context. And this does, I hate to tell you, this does happen. In a regulatory context, you basically have spelled out your analysis plan a priori. And you present that to the regulatory agency, be that in the United States, in Japan, in Europe, elsewhere, and you come to an agreement. This is the appropriate way to analyze this data, and that is the first analysis that you do. That is not to say it's the last or the only. You certainly can do other analyses, but you do the analysis that you had planned first. Okay? So those are some of the differences and similarities. You've learned a lot of the similarities. So non-inferiority. One basic difference, inherent difference, and unique aspect of non-inferiority trials and equivalence trials is that there is a pre-existing licensed product. So there is already a drug on the market that treats eczema, okay? And you are the drug developer of a new drug that treats eczema. And you're not interested in carving out a new niche for yourself. You're interested in showing how your product relates to the previously licensed product. So back to our shampoo example, Pantene and Head & Shoulders have carved out a market for themselves. They have spent millions and millions on advertising. Everybody knows how good they are, how great they smell, how well they clean your hair, and so forth. And so there is a prior licensed product on the market that you can sort of get onto their coattails, if you will, particularly when it relates to advertising and carving out a market niche. So there exists a prior licensed product. I can't stress this enough. That licensed product has a trail. It has a regulatory trail. It has usually has some trail in the medical literature. These days it has a trail on clinicaltrials.gov. It also has a trail in terms of reported adverse events. So you know a lot about this licensed product, or you can know a lot about this licensed product. You can know its effect size. How good does it work? Because there will have to have been a placebo-controlled trial for this product to get on the market, or a usual care controlled trial, or something like that that establishes the effect size for this prior licensed product. Again, depending upon how long it's on the market, there will be an adverse event trail, reports to the regulatory agency of the adverse events that are observed after licensure. So in addition to what you have in the pre-licensure phase for this product, you also have the post-licensure phase. So there's a lot of information on this pre-licensed product. And you have to know that information to design your non-inferiority trial. Now, another aspect of non-inferiority is the constancy assumption. And this is a critical one, and it's unverifiable. So you have to make an argument, make the case that the constancy assumption applies, that the situation in which you are trying or testing your new product against this prior licensed product is one in which the constancy assumption still applies. In other words, the effect that we saw in the original randomized control trial is still going to be the effect of this prior licensed product in our new trial that we conduct today. And the constancy assumption sometimes doesn't hold. And it doesn't hold if there are major seismic changes in the underlying medical care, in the underlying diagnosis, in the underlying disease process, in the concomitant medications, in all sorts of things. For example, think about if you were developing an antibiotic in the pre-penicillin era versus the post-penicillin era. Does the constancy assumption apply? I don't think so. What about if you were developing, and this is a current scenario, developing a new anthrax vaccine, and the licensed product was licensed decades ago? Does the constancy assumption still apply? You'd have an argument to make. The constancy assumption assumes that the underlying situation is the same in the current trial as it was in the original trial of the original licensed product. So in the era of antibiotics, things changed dramatically. In the era of heart, in HIV treatment, things changed dramatically. It's very difficult to carry the constancy assumption under those scenarios. If, on the other hand, you're developing a new eczema drug and the licensed product that you want to compare it to was just licensed last year, you probably have a reasonable case to make that the constancy assumption applies. But as you'll see as we go along, this is one of the burdens of the clinical trialists, the sponsors, when you're conducting a non-inferiority or an equivalence trial. You have to make the argument that the constancy assumption applies. And we'll go into that in a little more detail in a minute. The other thing that is different about a non-inferiority trial versus any other garden variety randomized controlled trial is that there is a lot of attention and effort put into establishing the margin. And this is a margin with a capital M. And I'll show you in a few minutes the FDA guidance document that talks you through the margin and gets into much more detail than I'm going to get into this morning in a short period of time. But this is one major difference between a placebo-controlled trial and a non-inferiority control trial where the control is the prior licensed product. I'm going to touch just briefly on switching. Sometimes you conduct a trial and you get really, really lucky. And your new product actually looks substantially better than the prior licensed product. And you really don't want to claim non-inferiority. What you really want to claim is superiority. And that's what we're talking about in switching. It's not switching the other way around, which is much, much, much more difficult and a much different scenario. It's switching from a non-inferiority claim based on a non-inferiority design trial when you have seen the results and now you want to claim superiority. So that's what I mean by switching. And equivalence. Equivalence is basically a tighter statement than non-inferiority. I'm claiming that we are not only not inferior to the licensed product, but we are equivalent. And again, that's a difficult thing to show. Sometimes you plan your trial to be an equivalence trial, and the design reflects that, and your plans for the trial reflect that. And I'm going to just touch on that briefly. Okay, this is the guidance that I talked to you about. Kay mentioned my colleague, Simon Day. Simon always gives me a hard time for how long it takes for FDA to come to any final conclusion on a final guidance document. You will see lots of draft guidance documents, and sometimes they're draft for years. And in Europe, they seem to turn them around in months. So, he has something to crow about. Anyway, this guidance document walks you through a lot of the detail, much more of the detail that I can cover in a seminar of this type. But it very clearly spells out what you need to do to design your non-inferiority trial, what you need to do to present the results to the FDA. And I commend that to your bedtime reading. It's a really good bedtime reading. I neglected to give you the URL, but it's available on the FDA website. This is a graphic that we use to show the difference between all the various trials that I've talked about already this morning. But let's start at the bottom here. This is the usual trial that you're used to, showing that some intervention, some test agent, as it's called here, is better than zero, better than often a placebo comparison. So if it's a placebo comparison and you are to the right of zero, as this confidence interval is, you can claim that superiority has been shown. because 0 is outside the confidence interval. These can all be, let's just think about these as all 95% confidence intervals. Okay, so this is the usual trial that we all are used to seeing and reading about in the medical literature. Now let's look at the various non-inferiority trials and equivalence trials. In order to assess the non-inferiority trials, you have to establish where these dotted lines are. And these dotted lines are the margin that I talked about earlier. Now, the margin is established by looking at the evidence for the prior licensed product. You have to delve into, fortunately, it's all available on the web now, and often slides that the sponsor presented to the FDA are available too. So establishing what this margin is, and the margin is called delta. Sometimes it's a capital Greek letter, sometimes it's a small Greek letter. But this is the margin that is discussed in the regulatory guidance, and we'll discuss it as we go along a little further and getting into reporting. This margin is sometimes exactly the effect size that's shown for the prior licensed product. That's in the regulatory guidance called M subscript 1. It is also sometimes the M2 margin, which is a margin that preserves a certain proportion of the effect of the previously licensed product. So the regulatory agency will say, you don't have to show that you have exactly the same effect as the prior licensed product, but you have to preserve at least 50% or 75% of the effect of that licensed product, or else we don't want to talk to you. So this margin, this dotted line, may actually represent 75% of the effect of the licensed product. But anyway, we've established what that margin is. The upper margin, which identifies the area shown in this graph where the test agent is better, and the lower margin, which identifies the area in this graph where the test agent, the control agent is better, and the sort of gray zone in the middle. So in the A scenario here, the non-inferiority is shown because this confidence interval does not cross the lower boundary here. Okay? So since this confidence interval doesn't cross this lower boundary, I can claim that my test agent is non-inferior to the control. Okay? So non-inferiority is shown in example A. In example B, oops, we crossed the line. Non-inferiority is not shown in that case. Does anybody have a guess as to how this design could have been improved to maybe show non-inferiority rather than miss the mark? What was that? Any guess? How do we normally tighten up a confidence interval like that? Okay, so the design of that study probably wasn't large enough to show non-inferiority. And maybe if they had done a larger study, they might have been able to show non-inferiority. But in fact, if they had done a larger study, they might have been able to show equivalence. The three in the middle, the three confidence intervals in the middle, do not cross these dashed margin lines. So in every case here, we can conclude that the test agent is equivalent to the control agent from a regulatory perspective. And that means what it says on the license for this test agent is this test agent is equivalent to that control. Okay? Yes?
So the upper one, I'm just trying to figure out the difference between equivalence and non-inferiority. Sounds pretty arbitrary, but the upper one is non-inferiority because it might be better?
It might be better, yep. There is a possibility that it's better. There's
Is that, I mean, is that the standard definition? Non-inferiority means it might be better. It's at least equivalent.
Yes.
It might be better.
Yes. It can't be worse. Yes. And this margin defines worse.
So non-inferiority actually suggests a higher level of confidence in the new product.
Mm-hmm.
But you didn't show all the possibilities.
Yes. Yes. There are multiple possibilities. But since we're talking about non-inferiority, these basically are the two that we need to focus on. Yes, in the back?
Yeah, isn't that a little bit misleading, the equivalence and equivalence not shown? Equivalence gives the impression, well, they're similar, but that fifth line, it actually, the confidence intervals tread over possible superior.
Mm-hmm.
You know, so the impression that you get when you say equivalence not shown is actually that it's worse when it actually is potentially better.
Well, I agree. This one deserves some additional explanation, but this confidence interval is not completely within the dashed boundaries.
But it's covering exactly the same areas of non-inferiority on the top.
On the top, yes. But if you design this as an equivalence trial, and this is what you got, equivalence is not shown.
Potentially
But
better.
non-inferiority is shown still. I mean, if you're using just the boundary,
Yes, you could.
that's exactly the same outcome.
Yes, you could claim non-inferiority. Yes, yes. But usually these are decisions that a sponsor makes with regard to how they intend to use this product, how they intend to market this product, what they want this product to do in terms of be a part of their larger portfolio. So they're going after something very specific here, and they wanted it to look like one of these three. Yes.
These terminology, inferiority, non-inferiority, equivalence, they've been with an FDA toolbox with companies, I assume, in the past 2 decades.
Yes.
They are recent, relatively speaking.
They're relatively recent, particularly non-inferiority is relatively recent. Yes.
Thought of that.
Yes. Yes.
So why do we need all those little fine-tuning of very confusing terminology from a regulatory perspective?
Well, from a regulatory perspective, if I were a product developer, I could just ignore the product that's on the market and just try to show that my product is effective against eczema, in treating eczema. Just ignore the marketed product altogether. All I have to do, and it's not a small thing, is to show that my product is safe and effective for what I say it can do. You can go that route. That is a different regulatory hurdle than going the route of showing that your new eczema therapy is equivalent to or non-inferior to this other eczema therapy that's on the market that has sort of undesirable side effects, costs 3 times what my product is going to cost. It requires freezers at minus 80 degrees, and my product can go in your backpack. There are a lot of reasons that a new product may be considered by the consumer better than the old product. And it's not necessarily the aspect of its biologic equivalence or how it reacts, how it demonstrates its effectiveness in a biological context. If a product is shown to be equivalent or even non-inferior, it may have lots of other aspects that are much more attractive to the consumer. The diabetes pens that are available now, the diabetic pumps that are available now, they have attractive features to the diabetic who used to have to have their insulin at the ready and don't have the ease and convenience of having a pen in their pocket. So there are a lot of reasons that you want to claim that your product is either equivalent or non-inferior. And it's usually easier to claim that your product is non-inferior.
Mary, I think
Yes.
it would be clearer if F said superiority not shown instead of equivalence not shown.
Okay.
I mean, I
Um, I'll have to.
really what it said.
Help me.
Yeah.
Okay.
So if it said superiority not shown.
Superiority not shown. Yeah, that's a good idea. Superiority not shown. And then we would have a little bit of symmetry here. So, okay. That's, I appreciate that because we're gonna teach this course in the summer, and you could check me on if this slide has been adapted. And I'll get to adapting in just a few minutes. Okay. So, understanding and being able to explain confidence intervals like this is a very important part of non-inferiority and equivalence trials. It hasn't been as critical with superiority trials like this because, as Tony Fauci once said to me, you could see that from across the room and you don't need a statistician to figure that one out, this one. Okay? All right. So I touched briefly on switching conclusions. There is an entire guidance document. Actually, it's called Points to Consider from the European Medicines Agency and from their CPMP, their Committee on Proprietary Medicinal Products. And this talks about just that issue of switching conclusions. Now, if you are not taking the results of your clinical trial to a regulatory agency, this isn't a critical issue for you. But if you are taking the results of your trial to a regulatory agency, you better read this. You better know what they're expecting and what are the limits of what you can do and what you can conclude. So that's the Switching Conclusions guidance document. And again, that's available on their website. I have to learn to put the URLs there.
Still on
Pardon?
prior hypothesis. I mean, is that just based, what's my primary endpoint, what's my prior hypothesis?
Yes. Particularly, what is your prior hypothesis? Yes.
I always thought it didn't carry a lot of weight anyway, but I noticed that.
Well, but it's already agreed upon with the regulatory agency. The data is the data. The data are the data. Yes.
I'll give you that.
You're absolutely right. The data are the data. But in a regulatory context, if you came in the door with a given hypothesis, with a given experimental design, with a given analysis plan, and after seeing the data, you're switching, that raises red flags. It is possible, and this guidance document outlines how to do it, and when to do it, and why to do it, and what needs to be done in terms of the regulatory submissions. But it's not as easy as it might be in a non-regulatory context. So yes, Kay.
Clarify something because there are students here and I want to make sure I'm right here. If you have a product where the product has gone through, let's say, the FDA for a particular indication, but you want to do a non-inferiority study for an off-label use, in a way, there's no such thing as switching because you're not beholden to anybody that you've switched from non-inferiority to superiority. You don't have to report to anybody that you did that, right?
If you're not seeking a license for that, yes.
It's already licensed for a different indication.
If it's already licensed for a different indication, but very frequently a sponsor is interested in a license for that indication, and maybe it's being used off-label in with another product in another context. It depends on what the sponsor is interested in. If they're interested in reporting the results of their trial in the New England Journal or the Lancet and letting medical practice take it from there, that's one scenario. If they're interested in getting a license that says this product has been shown safe and effective to do such and such, then they need to fulfill the regulatory requirements. So you have to know in which scenario you're operating. And switching scenarios, this is talking about switching conclusions from a single trial, but switching from the non-licensed, off-label world to a context of a licensed product is a difficult leap and requires a lot of effort. Okay, any other questions?
They don't want to show that. Like we said this and they've asked.
I think it is a difficult concept that we don't teach formally, probably, about what the FDA does have to approve and doesn't have to approve. So I think it's worth
Yes, you're absolutely right. And I do think that this is where I'll introduce my commercial message. Johns Hopkins has done a very good job in 2 courses specifically in introducing the regulatory concepts that work in concert with the concepts that you're learning in your other courses. They are very relevant to the regulatory context, but you need to know specifically the expectations in a regulatory context. So you can't just take what you did in your coursework at Johns Hopkins and go directly to Emergent or Roche or Merck or whoever and do the same thing. There is a regulatory level of rigor and demonstration that has to be there. But the principles on which all of these things underlie are the same principles as you've been learning in your other courses. Now, anybody who works in clinical trials knows that the best laid plans oft go astray. And to illustrate this is actually a parking structure at NIH that was not necessarily very well planned and fell apart as it was being constructed. So this is my example of best laid plans. The thing that we deal with most often in clinical trials that makes our best laid plans go awry is the issue of missing data. And one of the big distinctions between the regulatory, the pharmaceutical industry, for example, and studies that are being conducted with a plan for regulatory submission versus studies that are, say, NIH or CDC or WHO or somebody else funded. They may be exactly the same trial in terms of the hypothesis, the design, but there are differences in the conduct. And one of those differences that you'll see very strikingly is the effort to minimize missing data. And a pharmaceutical company will make great efforts to minimize missing data, particularly their primary endpoint. They will budget a lot of effort to minimizing missing data. They will introduce a lot of training of staff. They will do auditing. They will do a lot of things to minimize missing data. And they're very successful at minimizing that missing data. So that's one major difference. They will also, there is also the handling of missing data in the analysis plan. Missing data happens, you have to assume that it's going to happen, so how are you going to deal with it in your analysis plan? So your analysis plan, and this is what you discuss with the regulatory agency, are you handling missing data appropriately? Is this what we would recommend or what we would expect with regard to the handling of missing data? That needs to be spelled out in your analysis plan. And hopefully in many of your courses you're learning how to not only minimize missing data but analyze it appropriately. And one of the mechanisms that we have now that we didn't have years ago is imputation, multiple imputation. And there are a lot of great ways of dealing with missing data using multiple imputation, and we're seeing that more and more frequently. And that's the sort of thing that needs to be described in your analysis plan. Now, I think my next slide is, this is a relic of my days at the FDA. This is an award that I created and gave out liberally at the FDA since there was no monetary involvement, just printing out a color sheet of paper to give to people. I gave this to anybody that attempted to eradicate LOCF. Does anybody know what LOCF is? Yes, last observation carried forward. And there are permutations, BOCF and other permutations. Anyway, this is a horrible method. It's been denounced in the statistical literature many, many times. It's still appears in the medical literature. If you Google this, you'll find it. If you Medline search, PubMed search, you'll find it. Journal editors still let it in, let it slip by. If you search the FDA, there is an ancient FDA guidance document that still says LOCF is okay, but it's not okay. There are much better things to do, and it has serious problems. What happens is it's an easy thing to do. If you're doing a repeated measures design, and you see a subject monthly, and then after 6 months they're gone, you take the last observation that you observed on that person, and you use that value to fill in all the later measurements. Okay? That's an easy thing to do. It's a dumb thing to do. It affects the variance. It affects a lot of things. It introduces bias. It's just, and there are better things to do. So this is a dumb thing to do. And if you're very, very lucky, I will extend to you this award someday, and you can put it on your bulletin board, and you'll be among the elite. But do your best to eradicate LOCF. Remember, that is a mechanism for imputing with not a whole lot of effort or mental effort or computing effort to impute the value. So there are better ways to impute. Okay, now we get on to conduct and reporting. And anybody that works in clinical trials knows the scenario of garbage in, garbage out. We want to design and conduct trials that don't involve garbage in. And we want to also report and submit data that doesn't amount to garbage out. So that's what we're working towards. You're all, I'm sure, familiar with the consort statement. There is a specific consort statement that applies to non-inferiority and equivalence trials. And the reason there's a specific consort statement that applies to non-inferiority and equivalence trials is that there's more that you need to tell the reader. You can't just give the reader what you give the reader for an ordinary superiority trial. You have to give the reader more. And this consort statement gets into that. And here's a little brief run through of the checklist from the consort statement. You need to describe fully the participants in your trial, particularly the eligibility criteria, and the interventions, the control treatment. You need to describe that in sufficient detail that the reader knows how similar or not similar your participants and your interventions are to the prior trials. Remember, in a non-inferiority, superiority context, everything hangs on those prior trials. Even though it's like the elephant in the room. They're not the trial that you're conducting today, their history, but they affect the trial that you're conducting today. They have an impact. So the interventions are that control treatment, the new treatment, the objectives, clearly distinguish whether this is a non-inferiority or an equivalence trial, the sample size for your trial, including the margin. And I was hoping to have a really good example to give you from clinicaltrials.gov. And when I search non-inferiority, and you can do this yourself in clinicaltrials.gov, it's no good example forthcoming. Let me just leave it at that. So when you post your trials, your non-inferiority trials on clinicaltrials.gov, please do a better job than those that are already up there. Okay, the margin has to be described in your regulatory reports and also in your publications. That's how the reader knows if you've accomplished what you plan to accomplish. The other things that are in the consort checklist include allocation to treatment, the analytic methods, including the confidence interval, specify whether it's a one-sided or two-sided confidence interval. All of this, the confidence intervals I showed you on that previous slide were all two-sided confidence intervals. Sometimes one-sided confidence intervals are reported because you're really only interested in a non-inferiority trial in showing that you exceed the lower margin and you're not worse than the control. So sometimes you will see a one-sided confidence interval. The analysis sets are the intention to treat and per protocol, and I'm assuming that you've covered all this in various of your other trials. One difference between the ITT and the per protocol analysis set is that usually when you're doing a per protocol analysis, you have eliminated some of the non-adherers, non-compliant, you've eliminated people, and to get to your per protocol data set. That's a problem in equivalence and non-inferiority trials because that tends to move your treatment groups closer together. So it's a setup to show that they're equivalent. And so you have to be very careful in looking at both the ITT and the per protocol analysis data sets and analyzing as you had discussed in your analysis plan. And then you discuss your results, your baseline comparisons. This is very important so that you can show how your subjects enrolled in your trial might differ from the previous trial, the history that you're comparing to. And then you need to discuss protocol deviations, the outcomes and estimates of effect, and the interpretation. All of this is in that consort paper. And then we get to regulatory submissions. I probably have the world's largest set of slides of this kind of thing with pictures of stacks of paper. So if you ever need a slide with stacks of paper, I've got plenty of them. The kinds of things that you have to report to a regulatory agency are outlined in the Code of Federal Regulations here. And with regard to non-inferiority trials, it's important that the analysis of the study should explain why the drugs or devices or biologic products should be considered effective in the study, for example, by reference to results in previous placebo-controlled studies of the active control drug. So that has to be in your report to the regulatory agency. So again, you're not doing this study in isolation. The history always hangs over what you're doing and has to be a part of what you're presenting. I did want to indicate that the regulatory process is an evolving, moving process, and things change over time. And here's one good example of things changing over time. This is a workshop that was held several years ago that actually Johns Hopkins was one of the co-sponsors of this workshop. And you can, I think I actually have the URL for this. You can find all of the information on this workshop in these two URLs. But that's an example where the regulatory process is evolving, has been evolving, and is evolving. Another example is in adaptive designs. I'm sure you've all heard of adaptive designs. But as you can see here, there really is no universal definition. You can adapt a lot of things in your trial. You can adapt the eligibility criteria. You can adapt the sample size. You can adapt the duration of your trial. Lots of things. There's lots of new methodology being published and still published today. And there is also regulatory documents that are available on adaptive designs. There are multiple terminologies such as flexible design and attractive design. We would all hope that all of our designs are attractive designs. And then PhRMA, which is the Pharmaceutical Industry Association, has its own definition, and that is what you see here. An adaptive design is a clinical trial that uses accumulating data to modify the design of the study, and it continues, and that modification occurs without undermining the validity and integrity of the trial. Here's the trick to do that. Okay, so this is another one of those evolving scenarios. And what I'd like to encourage all of you to do, what I saw when I was working on the regulatory side of life, was sponsors coming to the FDA, and I can only speak from the FDA because that's the only agency I served in. The sponsors would come to the FDA, and they would not want to push the envelope with regard to experimental design, with regard to analytic methodology and so forth. They would want to toe the line and stick with what has worked in the past, which is why we are all stuck with LOCF. So what I would urge you to do is to take what you've learned at this august institution and take it out into the field and argue the point that this methodology that has just been developed in the last 5 years is a better methodology than that which has been used for the last 30 or 40 years. If you're not willing to make that case to the regulatory agencies and to the pharmaceutical industry or the sponsors of any type, then the regulatory process will come to a halt. It will not evolve. It will not progress. And you are the future of that. You need to make it happen. Yes?
Is there a standpoint from the FDA regarding these adaptive designs? Do you have a feeling what they think about these designs?
There actually is an FDA guidance document on adaptive designs. It indicates which approaches have more information, and we have a comfort level that is higher for these approaches. And it indicates these approaches are new ideas that have yet to be vetted and well documented, and we're still a little nervous about these. That's what the guidance document says. So if you are developing an adaptive design, you really better read that guidance document. If you are planning to publish your dissertation on adaptive designs, it wouldn't be a bad idea to read that guidance document and to adapt some of the approaches or language and methodology and context that you see in that guidance document, and use that to craft your dissertation topic or the way you convey your dissertation results, for example. So there are a lot of ways that you can help the regulatory process evolve. And I look forward to seeing you do just that. I would like to have some time for questions, but I'd also like to say that I'm sure all of you who are involved in clinical trials agree with this cover from Science, that clinical trials are trials and tribulations, but they're also the most fascinating and interesting and challenging, and that's the reason that we all get up in the morning. Thank you. Any questions? I hope you all look, when you're shopping in the grocery store, look at all those equivalent products. Yes?
Mary, thank you very much for that. This whole thing about what should be reported and where it should be reported, and the fact that the clinicaltrials.gov, when you were looking for sample size, indicated nothing about margin, sort of wasn't surprising to me because, there's no specification that margin be reported there. But what you do want to report in the regulatory and as well the control statement, is specifying it for publication. How would you think about going about changing it so that what's required is consistent, across sources?
Well, that's very hard to do. It's hard to do in the context of the medical literature because not everybody buys into the consort statement, even the first consort statement. It's very hard to do in the regulatory context. There is an effort, and it's about a 15-year-old effort now, to harmonize across the major regulatory agencies of the world, Japan, Europe, and the U.S., and that's been going on for 15 years to try to harmonize what everybody's doing. We can't even really agree on how to spell harmonize, so we haven't gotten very far. So I think it's an uphill, and I would actually rather see good practice evolve from the bottom up rather than have some journal editor who, once they're gone, the next journal editor may think differently. I think what we cover in the courses that we offer here is training in what is good practice. And hopefully, when you are out in the practice of clinical trials that are intended for regulatory consumption, you will remember these good practices and not get bowled over by somebody from marketing who wants to get it done faster and cheaper and sloppier, and just don't give in to them. And that's a stage much, much earlier than what the public sees. There was a question over here? Yes.
Yeah, thanks. Well, first, Maria, I have a comment. I was surprised that clinical trials that didn't require that because of all the nonsensical statistics that are required.
But it means they have to say, and for this type of trial, margin.
But, I was thinking in terms of a lot of times when we're doing superiority trials, we end up with negative results. And so we have these kind of negative trials that we publish a lot and actually sometimes make a big to-do about our negative trial. And I'm wondering if there are ways...
No, you don't publish negative trials.
She just did. In general, thank you very much.
She's our star.
Anyway, shouldn't we try to use some of the principles in the design and kind of discussion of non-inferiority trials in presenting those results?
Mm-hmm.
And it would just, maybe even, and I just wanted your comment.
Right. Well, yes, I agree that if you're going to publish something that was attempted to be a superiority trial and doesn't show superiority, the onus on you as the investigator is to present what can be learned from this trial and what is positive from it. We did learn that this and this and this. We may have a confidence interval that's wider than we would have liked. We may not have shown rigorously that this product is better than that product, but we have shown the following. If your example has already passed the review of the journal, there must be something substantive and new in what you've done. Otherwise, it wouldn't have been published. So, I think the onus is on us as the investigators to write clearly the results, and it's a hard thing to do, to write the results so that the next investigator, or think about if you're the next patient or the next parent of a patient who is making the decision, do I use this product or do I use this product? They need information, and that is what the clinical trials process provides. And it doesn't provide sound bites, it provides information, which is not consumed as a sound bite. It's consumed by a thoughtful person trying to absorb the entire package. So, it's tough. And actually, our whole society is geared towards sound bites. And the more we encourage education of not only clinical trialists, but consumers in general, for them to appreciate what a licensed product versus a prior licensed product versus an equivalent or non-inferior product, what that really means. I think that we still have a long way to go. Does that answer your question? Yeah. Okay, thank you. Yes.
Thank you so much. This is a wonderful talk. How do you relate this work, regulatory maybe perspective, to comparative effectiveness research, maybe down from CMS,
yes,
HHS?
Mm-hmm.
How do you see them relate to each other, or how they can be harmonized in terms of my teaching, for example?
Yes, that's a very good point, because until recent years, in a regulatory context, you would not see one product from one sponsor compared to another product from another sponsor. But that is exactly what's happening in comparative effectiveness. So the information that's given out may actually be sufficiently rigorous. The information that comes from comparative effectiveness may, in fact, be sufficiently rigorous to go through the licensure process. If you're talking about two licensed products, which oftentimes we are, and we conduct a comparative effectiveness study to compare the results in the real world of those two licensed products, again, you present the consuming public with the results of that trial. You present the level of detail in the comparisons, the head-to-head comparison. But the consumer then is left with, I may have aspects that are much more important to me that you didn't put much emphasis on in your study, and that may be one particular adverse event that is just unacceptable to them, and that makes the decision. But they also, on the basis of the comparative effectiveness study, know that the two products have been compared head-to-head, and with regard to clinical efficacy, they are essentially comparable. Maybe not shown as rigorously as a regulatory agency would require to put that on their label. And if they're two licensed products, they're not really going to be interested in that on their label anyway. But I do see comparative effectiveness as sort of a continuation of this these sorts of comparisons, and I hope the kind of rigor and detail and level of information that I've touched on here goes to the way that you might report your CER studies, so that the consumer gets all of that. Yeah, I agree with you completely. It is along the same path. I agree. Do we have time for one more? Two more? One more? Yes.
I'm still thrown off with this non-inferiority versus equivalence. I mean, I understand the difference. I'm trying to understand exactly why somebody would want to do an equivalence study rather than a non-inferiority study, because it would involve a larger sample size, so you can have tighter confidence intervals. And then, if their product is better, then they can't say it's equivalent. Whereas with the non-inferiority, they can. Is it just the connotation that equivalence, marketing something as equivalent, better than marketing something as non-inferior? Which,
yes,
though it's non-inferior, that gives some connotation that it is inferior.
Yeah.
Isn't that just by putting that word out there?
Just by putting that word out there, that may play into the decision on the part of the sponsor. I don't want to go for non-inferiority because I cannot sell that word non-inferiority. And so they go for equivalence.
Didn't you say at the beginning when you first came with the ship, I never heard of it before, that it said it was the same or as effective as? They didn't say and you said that's with non-inferiority. So it doesn't sound like if you do a non-inferiority trial, come up with non-inferiority by your definition, and somebody has to say that.
No, they don't have to say that in their commercial, dancing off into the starlight. They don't have to say.
Life very complicated.
Well, I hope that I've at least cracked the confusion that you're trying to describe here, because it is a confusing scenario. But if you are a sponsor, you make that decision. Do I go for non-inferiority? Do I go for equivalence? Do I go for superiority? The sponsor makes that decision. And if you're sitting in the chair of the sponsor and you want to make this your blockbuster drug that sells billions over the next decade, you probably want to go for superiority. And if you've got evidence in your preclinical studies and so forth, you might want to go that route. If there are indications that, it'll cost you $80 billion more than this drug will ever make to get an equivalence claim, you might go for non-inferiority. You have to make that decision as the sponsor. Can I talk to you afterwards? Okay. Thanks.
Thank you very much.